He then went on to do his postdoctoral work at the Yale Medical School, serving as a research scientist for 7 years. On his return to India, he worked at the Tata Institute of Fundamental Research for several years, being a senior professor and chair of biology. His areas of expertise are the molecular basis of genome dynamics, computational biology, and genomes and protein active sites, cell physiology, and metabolism, areas in which he has made fundamental contributions. In addition to his research in the field of biology, he is also interested in dissemination and public relations of scientific knowledge. He has won many awards and grants. He worked as his DST Inspire teacher, bringing the current excitement of basic biology to students across India, and has given numerous technical and public lectures to various audiences. We will now invite Professor B.J. Rao onto the dais to give his talk on the topic, What Makes Us Uniquely Human? Lessons from Our Extinct Relatives Through Paleogenetic Analysis.
Firstly, thank you to the Young Scholars Club for doing a splendid job of organizing this series. I believe this is the first offline series which is working extremely well. Now, I'm trying to cover this year's Nobel Prize in Physiology and Medicine.
At least this guy remembered our...
The citation actually reads the following. For his discoveries concerning the genomes of extinct hominins and human evolution. Svante Paabo. The purpose of this presentation is to convey three things. One, why topic given Nobel recognition, number one. Number two, who did this and who was he? These are the issues that we want to sort of understand. Now, Paabo was a Swedish. He was born in 55, obtained his doctoral degree from Uppsala, conducted his postdoctoral work in Zurich as well as Berkeley, joined as a faculty at Munich, and then subsequently went on to establish Max Planck Institute for Evolutionary Anthropology, where he was invited to head the program. So this is him. And actually, what was he? What did shape his work? People go on doing their work because of certain things that happen with their lives. He was deeply interested in natural things, natural material, historical material, all the material that happened past, deep past. So he was interested actually in looking at Egyptology, all the relics in Egypt, such as mummies and other things like that. He thought that Egyptology is a very, very exciting area of science to learn about our deep past. Things like that at the University of Uppsala, but got somehow disappointed by the way Egyptology was taught there. It was not as romantic as I thought it would be. These are his words. And after a year and a half or so, I did not know what to do. Many Nobel winners actually go through this phase. They don't know what to do at some point because they are searching for something that excites them. Because this wasn't really it. The Egyptology was really not it. So I started studying medicine because I figured I would get a profession and it also will give me a way into basic research. But he ended up doing something different. He of course joined medical school. He of course joined a PhD program. But at the end, what he ended up doing was very different. That very different actually connected to the Egyptology spirit. What was that? The current setting says the following. Our lab pretty much functions on the ideas that are born in the group. This is related to his current activity in Max Planck. And our lab is a little unusual in that we spend a lot of time discussing every project every week in a group setting. All the groups that have to do with ancient DNA, he's still going back to the Egyptology spirit. All the people sit together once in a week and discuss their work, particularly things that don't work. Gene expression in another day, genomics, and it is in these sorts of eternal discussion that ideas come. The last few sentences are very remarkable. It is quite rare that anyone sitting in alone, thinking in their own room, comes up with any big ideas. It's very rare. It's really by throwing lots of ideas around. If you have 100 ideas on the table, then one of them turns out to be really cool. This is what happens in real world when you want to do something very exciting. And in fact, this is what happened in Paabo's world also. So Paabo's milestones. In 2010, he succeeded in sequencing the genome of Neanderthal. We'll explain what is Neanderthal. He also discovered a previously unknown hominin, Denisova. He also found that gene transfer had occurred from these now extinct hominins to Homo sapiens, that is us, following the migration out of Africa, which is about 70,000 years ago. The gene transfer made us bits and pieces of early hominins. We are all early hominins in bits and pieces, which ended up shaping us as what we are today. This ancient flow of genes to present-day humans has very high physiological relevance, affecting how our brain and immune systems work, two most important systems. So you see there is a lot of insight here. These are hominins which became extinct. Before they became extinct, they transferred genes to us. Those genes are shaping us now. Therefore, it's a very important discovery because it is telling us who we are and how did we come about the way we have come about. Some basics let's look at. This is the evolutionary basics. You can see we are here and we are immediately connected with our hominins here. Now we are actually earlier, before hominins came along, we were connected to chimpanzees, gorillas, etc. Chimpanzees, gorillas are the living nearest cousins to us. They are here now. But our real nearest cousins became extinct, which are hominins. This is the essence that came out of his work, and if I just blow it out, this is how it is. We are us, our nearest cousins in the genetic terms, Neanderthals became extinct, but before they became extinct, we and them interbred some 40,000 years ago. These are real numbers. And this interbreeding did lot of good to us and some bad to them so that they became extinct. We'll explain what that is. But before they came along, our best known nearest living cousins were chimps and gorillas. All this branching started some 9 to 6 million years ago. These are primates and these are also primates. So you can see that we are talking of long past here. And long past is thereon by two important techniques. One is carbon dating. Carbon dating cannot go too far. It only can go 50,000 years, for that duration only, because the technique has uncertainty. But this long time scale that geneticists talk about has to do with how genetic mutation rates happen. There is a genetic mutation rate in a neutral evolutionary theory, and by the genetic changes that happen across these species, you can actually compute how far ago they branched away from us. There's a method, robust method, that computationally you can assess using genetic sequences available in all these sequences, in all these animals. You can go back and see, oh, this sequence is so far different from me, so it has to be so far away in terms of time scale. So these are the approaches people use and try to figure out who is my nearest cousin evolutionarily. And then the next question is, how am I different from my nearest cousin? And why am I different from my nearest cousin the way I am different? These are somewhat fundamental questions, because this is like asking about ourselves as to how we are and why we are the way we are. So therefore, this prize actually deserves Nobel. Now, sequencing all these extinct species is a big challenge. Why? Because this is like sequencing a needle in a haystack. You don't get enough material, right? If you have to sequence human genome, it's very easy because we all can collect human genome very, very easily, pristine quality you can collect. But if you were to look up a dust particle sitting on this and sequence that with high quality, high certainty, it's a challenge. But current modern genetic technology does allow us to amplify it, and then computationally you can filter it, and computationally you can purify sequences and say with certainty, yes, this must be Neanderthal and not human. There are techniques available like that. So new technology was invented largely by Paabo for this purpose. Before he applied this technology, we will not go into the technicality of these new technology because that's not the purpose of this. We want to know what he did, whatever he did with the technology. Before he applied this technology to discovering new species, which is a big thing, he actually used this technology for testing on many, many innocuous material. He retrieved samples, very rare DNA samples from all such material. He was an Egyptologist by mind, therefore he looked at DNA sequences from all this material. So his original spirit was still alive, to do something about the past and then relate to current. When he studied Egyptology in Uppsala, the way it was taught did not bring him out that this is not what I want to do. But the spirit being there, he realized that genetic technology is evolving so fast, such high throughput, high quality techniques are coming along. So then he realized that I should be able to use this technology and do what is called paleogenomics, DNA sequence analysis, and using that data, high throughput data, I should be able to connect myself to the past. That is the original genesis. I think that genesis is what made him what he became. Not many people have this kind of genesis. Everybody wants to study what is happening to my life, how to improve my life. He was not interested in that. He wanted to know how deeply we are connected in the past. So now let us go into the challenges of what he did. Seemingly impossible task. If you had discussed with biologists in 1990 or so, during that time, nobody would have thought that you would be able to sequence an ancient, rare relic. It was an impossible task. No sensible geneticist would say, please do this project. No. There should be something inside for you to say, no, this is what I will do. So in 1990, Paabo continued his work on archaic DNA, where he decided to analyze DNA from Neanderthal mitochondria. Mitochondria, why? Because before you sequence nuclear genome, it's easier to sequence mitochondrial genome because there are multiple copies of mitochondria, number one. Mitochondrial size is much smaller than nuclear size. Easier system you'll handle and learn. The mitochondrial genome is small, you have thousands of copies. So he refined the methods. Paabo managed to sequence a region of mitochondrial DNA from 40,000-year-old piece of bone. This is where the journey starts now, the real journey. Thus, for the first time, we had access to a sequence from an extinct relative of ours. Now, little bit of a background of how sequencing is done for those people who don't know what sequencing is all about. If you take this as our chromosome or any chromosome, sequencing is done by a brute force method. You simply chop up the DNA into multiple random bits. You do what is called shotgunning. You actually take a gun and shoot it at near range. So you splatter the DNA into multiple pieces randomly. Each piece is sequenced. Since the sequence is maintained by continuity, there will be overlapping fragments in random collection. Computationally, you can stitch them, and then you can regenerate the entire sequence of the chromosome. You shotgun them, you sequence bits, and you can sequence these bits at a very high depth. You can sequence at 100x, 200x, 300x. So your quality of sequence will be so high that you can filter out sequencing errors from real sequence. These are all computational. Once you do this, you can actually assemble high quality, 99.99% fidelity quality into a full-length chromosome. That can be done. This is good in terms of sequencing technology, but the challenge is that when you take DNA from a relic sample, there will be a lot of fresh DNA coming from microbes, there will be fungi going. DNA sequences will be seriously underrepresented in the way you do as per this method. Correct? Populationally, they will be seriously underrepresented, and therefore, underrepresentation is a problem, but the quality of underrepresented fragments must be of high. Then there is a way that you can sort it out. So therefore, you can take those underrepresented but high quality sequences, align with the human genome, because human genome is the model template. We want to know how far we are away from them, right? That's the original question. So align with them and ask the new ancient DNA, is it related to us? Yes, they are, because you can align them sequence-wise. You can do it with high throughput computational methods. Ultimately, you will realize that yes, the sequence that is present in the ancient DNA is significantly related to human genome in so and so loci. This is the answer you will get, unambiguous answer. Then that is when the real journey begins. While this struggle was happening, serendipity also helps people who are genuinely working. So they came across very good quality archaic genomes, because there were caves which were very cold. DNA quality was much better preserved. The samples that they got in these caves were much better, higher quality than what they were earlier used to. So therefore, the material itself improved when their methodology was getting improved. At the end of it, they discovered that there are Neanderthals in this part of the world, Western Europe and Western Asia, and another hominin called Denisovans in the eastern part of the Asia. Samples were retrieved from multiple areas of the Europe and Asia, and this is how the distribution looked. These sequences are distinct from these sequences. Both these sequences are in certain regions distinct from our sequences. And this is the timing of the situation 500,000 years ago. This is the time zero of the system now. Out of Africa, humans did not still come out. It's still a question mark. We are to come out later. So that means these are our early cousins who actually came out and settled in this portion of the world. They were ahead of us in the out of Africa migration. Before these hominins were discovered, the notion was that early human beings were the first to come out of Africa. Not so now. They came out of Africa before us, and they populated in Europe and Asia, etc., in this kind of distribution. This is 500,000 years ago, and later, 50,000 years ago or so, modern humans, the so-called us, started coming out of Africa. This is what we had known earlier as time zero, but this was actually the time zero. Now, when you come out and say, oh, there are already people like us who actually can speak like us, there is evidence for that, based on the jaw bone structure. Bone structures can be simulated for those animals who could speak versus those animals who could not speak. This is a very important, serious area now. Then they realize that there are animals, there are hominins who are actually speaking, who have their own articulation, and then the modern humans, the so-called modern humans came along. There was enough interaction between them, and then obviously when there is enough interaction, there will be genetic matings, which is what happened after 50,000 years later, when modern humans spilled over into out of Africa, into Europe and Asia. Now, some details. What all did this group analyze? Neanderthal genes survive in us. In some of us, they live on a little bit, said Professor Svante Paabo from the Max Planck Institute. And the study came from researchers looked at 5 groups of modern humans from French, Han Chinese, Papuana, Yoruba, San. All these different groups were looked at. These are already current sequences, and these sequences which were retrieved by computational methods, these were compared with these groups, and by the genetic relatedness, this branching was generated. Okay, so now finally what comes out is if we analyze ourselves sitting here, each of us, we will pick up Neanderthal sequence like this. This is in one individual, this is in another individual, another individual, another individual, etc. So if you now add up all these, at each individual level, we are 1 to 2% sequence per person of Neanderthal. And if you add up all these individuals, we become in toto, in a populational level, about 40% of Neanderthal in us. At individual level, 1 to 2%. And we know exactly where we are in Neanderthal and where we are human. So we know the genetic loci where we are Neanderthal and where we are human at a very high precision level in terms of sequence. So sequencing the Neanderthal genome, comparative analysis showed that the DNA sequences from Neanderthals were more similar to sequences from contemporary humans originating from Europe and Asia than to contemporary humans originating from Africa. This essentially shows that horizontal transfer happened directly, and therefore they are more closer in terms of sequences. This means that Neanderthals and Homo sapiens interbred during their millennia of coexistence. In modern day humans with European or Asian descent, approximately 1 to 4% of the genome originates from Neanderthal. This is a hard conclusion based on high quality sequence data and very highly reliable computational profiling. So sequence of genome, at this time he was offered the chance to establish Max Planck Institute at Leipzig, Germany. At the new institute, Paabo and his team steadily improved the methods to isolate and analyze DNA from archaic bone remains. He also engaged with several critical operators, the key thing, with expertise on population genetics and advanced field analysis. None of these by training himself, but he put in people together. He even put in anthropologists, sociologists, because he wanted to understand how the Neanderthals behave based on sociological perspectives, anthropological perspectives, etc. So there is a holistic understanding that he's trying to establish at this center. His efforts were successful. Paabo accomplished the impossible and could publish the first Neanderthal genome sequence in 2010. Now the outcome is the following. All these Neanderthals, and one more